Hello everyone, my name is Hanan Azabi. I'm a bioinformatics student and today I'll be talking about ChIP technique. ChIP stands for Chromatin immunoprecipitation. Before we start, let's talk about some concepts we need in order to understand ChIP technique. Let's start with the chromatin. Chromatin is a complex of DNA and proteins, and it forms the chromosomes within the eukaryotic cells. Immuno refers to antibody. The antibodies are special proteins your body produces to protect you from disease factors. The most important thing about antibodies is that they are highly specified to a specific antigen, and fortunately we've found ways to produce antibodies in the lab. So, here's the case. Let's say we want to study a specific protein. We can artificially produce antibodies for that protein. These antibodies can selectively bind to the protein of interest. This technique is called immunoprecipitation. Okay, we have this protein sample. We want to study the red protein, so we have to isolate it first. We add to the solution a specific antibody against this specific protein. The protein and the antibody will bind together to form a complex. Then we have to get rid of the green proteins. We have many ways to do this. We can use magnetic poles, or such in this case we used another protein that makes the complex heavy and insoluble. So this complex will sink into the bottom and then we wash all of other stuff out of our solution. One thing that matters is that proteins can be binded to other molecules, such as another protein, DNA or RNA. In ChIP technique, we use immunoprecipitation to study the interactions between proteins and the DNA in the chromatin. These proteins might be histones or transcription factors. Let's say that we've identified a new transcription factor, yet we don't know which gene does it regulate. ChIP technique can tell us the answer. These are the steps we need to do. First, cross-link the chromatin. We usually use formaldehyde for that. Then we cut it down onto smaller fragments using sonication methods, nuclease, or enzymatic digestion. This fragmentation is necessary to solubilize the chromatin and to prepare it for the next step, which is immunoprecipitation. As I've already mentioned, we use specific antibodies against the protein we want to study, which is the transcription factor. Then we reverse the cross-linking and the DNA fragments will be purified, and finally we can do our DNA analysis using different techniques such as PCR or sequencing. ChIP can also be used to study the interactions between DNA and histones or DNA and histones modification. This was my topic for today.